What if you leave a sandwich in your bag for a long time? A green to black color would appear on it. But what does this color mean? It means that your sandwich has microbes. Where did these microbes come from? Do you see all these questions you have? Scientists in the past had as well. They actually conducted many experiments to know from where living cells come from. They believed in something called spontaneous generation. The idea of spontaneous generation suggests that life can be made from non-living material. We have mentioned before in the cell theory lesson that cells can only be formed from pre-existing cells. The microbes in your sandwich came from pre-existing microbes that were found in the air or even in the plastic bag. So how cells make new cells? There is only one way, and that is by cell division. In case of prokaryotic cells, new cells are formed when prokaryotic cells divide by binary fission. On the other hand, eukaryotic cells divide by mitosis to form new identical cells, and by meiosis to form sex cells, which are the eggs and sperms. The theory that cells divide to form new cells from pre-existing cells replaced the theory of spontaneous generation. In the past, many scientists conducted experiments to disprove the theory of spontaneous generation and support the idea that new cells come from pre-existing cells. One of those scientists was Louis Pasteur, who was a French chemist and microbiologist. He designed an experiment to test whether sterile chicken broth could spontaneously generate microbes. His experiment was called the swan neck flask experiment because the flask he used in the experiment had a swan neck, a curved one. In his experiment, Pasteur prepared two setups of swan neck flasks. He added chicken broth to both flasks and then he boiled the two flasks to ensure that all existing microbes were killed. In the first flask, the neck was broken off and the broth inside became contaminated. However, in the second flask, the curved neck of the flask prevented air from contaminating the chicken broth inside, and therefore the broth remained sterile. From this, Pasteur concluded that microbes do not spontaneously generate, but rather come from pre-existing microbes, which means that life makes life. Pasteur's experiment provided evidence that new cells come from pre-existing ones, and this was a great discovery at that time. Even Darwin's theory tells us that all living cells came from one living ancestor. Yet, we still have a very important question. How were the first cells formed? Those scientists disproved the idea of spontaneous generation, but they still wondered, is it possible that the simple life forms could come from non-living matter? Many scientists believe that the first cells came from inorganic compounds, which are non-living material. Until now, there is no evidence that completely supports this theory. However, two scientists, Stanley Miller and Harold Urey, conducted an experiment through which they were able to stimulate the conditions of the Earth and make simple organic compounds from inorganic compounds. Let's see how. Miller and Urey stimulated Earth conditions in a closed container. Water vapor was passed through a mixture of ammonia, methane, and hydrogen. These compounds and elements resemble the early Earth atmosphere. Electricity was then added to stimulate lightning discharge. As a result of this, amino acids and carbon compounds were created. This experiment provided an evidence that inorganic material such as ammonia, methane, and hydrogen can be changed into organic matter, such as amino acids and carbon compounds, under stimulated Earth conditions. Based on this, scientists believe that the first cells in life came from non-living matter. The origin of life is still a big mystery in the world of biology, and until today, we still haven't found answers to many of the questions or wonders we have. Maybe though, one day, the work of scientists across the world could help us reach some answers.